Hello, and thank you for taking the time to join me for this presentation. My name is Emily Guertin, and I am a doctoral student at Brock University's Child and Youth Studies Department, and I will be presenting my recent work on how GARRE1 predicts longitudinal reading achievement across 4 phonological and reading skill constructs. Before we launch into our study, I should indicate the disclosures of my co-authors. Each of the co-authors on this project are listed as co-inventors on the provisional patents listed. In this recorded presentation, I'll talk about heritability estimates for longitudinal reading skills, in addition to the molecular genetic approach to reading and our discovery of GARRE1. I'll also provide information about our study and the overarching results. If you have any questions about my study or its implications, I invite you to join me for a live question period on Thursday, July 15th at 3 p.m. BST. Because I am going to talk about several different outcome measures, I thought I would start our talk by briefly structuring how we are looking at the outcome measures of reading today. This is just an illustration, but we can think of a continuum of pre-reading and component skills to functional reading with comprehension. The idea is that children are first learning these component skills, then learning to read words, gaining fluency, ultimately with the goal of learning and functioning well in contexts that require reading in daily life. We know that reading, even longitudinally, is highly heritable. We learn this from behavior genetic approaches using twin studies, looking for monozygotic and dizygotic twin differences in reading achievement over time. These studies provide a global estimate of the overall contribution of genetics to reading and reading-related skills. H squared represents the variance in reading ability that is accounted for by genetics. On this slide, there are time point estimates of heritability in longitudinal studies. This means that at any given point in a longitudinal study of children, we can see ranges of heritability estimates from a lower proportion of 0.14 for phonological awareness, all the way up to a high proportion of 0.84 for word knowledge. There are also studies that have looked for the heritability of growth and stability in reading over time, and these studies show a global genetic effect of the stability in reading scores over time, meaning that there's a genetic effect on the relationship between a child's reading scores between 2 or more assessment points. We also see global genetic effects in the rate of reading skill acquisition, so the slope of a trajectory, and deceleration from latent growth models. This means that there are genetic contributions to both our rate of acquisition of skills and the stability of our reading ability over time. Molecular genetics add to behavioral genetics by looking for specific genetic variants that contribute to developmental dyslexia, reading ability, or cognitive components of reading, such as rapid automatized naming, or RAN. One approach is to look at families with frequent cases of reading disabilities and then sequencing the genes to find candidate genes for the phenotype, though these often yield rare variants that occur infrequently in the population. A genome-wide association study, or GWAS, of reading ability takes a reading phenotype and looks for associations with genetic information. This analysis also includes a severe correction for the number of tests conducted within the analysis and may result in the discovery of more common variants in a population. Several candidate genes and loci have been found and replicated for reading, and I have listed them here. We conducted a GWAS recently within the New Haven Lexinome Project, a longitudinal study of children of primarily Hispanic and African American descent. Their reading skills were measured from the beginning of grade 1 to grade 5. In this GWAS, a latent variable of decoding level reading was used to look for genetic associations. 31 SNPs were found meeting significance, resulting in the discovery of RS2559953, now referred to as GARRE1. The purpose of this study was to look at the longitudinal role of GARRE1 in 4 main reading measures in the NHLP population. We looked at the relationship between GARRE1 risk variance and both raw and standard outcome scores, in addition to both intercept, which means the differences in initial performance at their first test in grade 1, in addition to differences in the rates of skill growth, so their slopes of their trajectories. We also wanted to account for domain general, cognitive, and environmental predictors that were relevant to the reading skills of our population. To briefly return to this continuum of reading skills, we discovered GARRE1 using a decoding and word reading latent variable, but also wanted to look at the breadth of this relationship. So going down to component pre-reading skills all the way up to reading comprehension, and determine if the effect of GARRE1 was robust to other important factors in reading development. Sorry for my dog sneezing there. Our New Haven Lexnome project, as I described briefly before, measured reading skills at the beginning and end of each school year between grades 1 and 5. We also measured cognitive abilities and IQ in the middle of each school year. We had 420 children from 24 schools in New Haven, Connecticut, of primarily African American and Hispanic descent. A subset of these students received 2 tiers of reading intervention, reading recovery for 1 year or Empower for up to 2 years, for a total of up to 3 years. The predictors of interest in our growth models were child biological sex, their socioeconomic status, as measured by the report from parents of receiving forms of government assistance, language exposure other than English in the home, performance or nonverbal IQ from the WASI, auditory attention from the NEPSI, and some genetic variables from our sequencing data that account for genetic background. Our outcome measures included raw score subscales and composite standard scales of the Tower, Woodcock-Johnson, CTOP, and GORT for a total of 18 outcome measures and 18 growth models. I used hierarchical linear modeling, or HLM, to fit basic growth curves, comparing factors like the measure of time using the AIC and BIC to determine the best fit. We used a time variable of age in months of the child centered at their first test date in the study. We nested by enrollment school for all outcome measures except for the subscales in the CTOP2, where it did not indicate a better fit. All measures showed a deceleration term in random effects except the phoneme isolation measure and the RAND measures from the CTOP. All raw score measures had a deceleration curve in the fixed effects except for the RAND measures, and standard score fixed effects were best modeled with just a linear term. I would be happy to discuss the modeling process further if you have any more questions about this process. Here are the results of the covariates on the initial reading performance in grade 1. Each asterisk color denotes a different predictor, with the legend at the top of the screen, and asterisks indicate a significant effect of this factor for that measure. Of note, performance IQ was a significant predictor of all outcome measures except for RAN, and socioeconomic status and the completion of the intervention were also significant predictors. To show an example of the intercept predictor effects, I have a graph here of the Woodcock-Johnson passage comprehension raw scores, with groups split by socioeconomic status. The shaded regions indicate 95% confidence intervals for the mean. The red group did not indicate that they received government assistance. So in this context, that is interpreted as a higher SES group. And the teal group did indicate receiving government assistance. So that is interpreted as a lower SES group. Those with lower SES performed consistently lower than those with higher SES throughout these growth curves. We also tested if the covariates predicted linear growth of reading skills, and that means did they predict the slope of their growth. Evidence for other languages spoken at home was a significant predictor of overall steeper and faster growth in a number of measures, in addition to some effects of sex, socioeconomic status, performance IQ, and auditory attention. Here is an example of the effect of language exposure beyond English in the home, showing a steeper rate of acquisition than those who only had evidence for English spoken at home. The teal group indicates the group who had other languages spoken at home, and the red group, again, sorry for the dogs, indicate the group that only had evidence for English. You can see that the groups start off largely overlapping, and then the Other language exposure group at home shows a faster rate of acquisition in the second half of the growth trajectory. Our results for GARRE1 indicated that GARRE1 risk variance predicted lower reading and phonological skills in 17 of our 18 outcome variables that we assessed. The only exception was the CTOP2 phoneme isolation. We found one instance of GARRE1 predicting growth, such that GARRE1 had a higher slope and showed converging slopes with the non-risk group over time. I have some examples of these effects. In each graph, teal represents the GARRE1 risk group, and red, perhaps counterintuitively, represents the group without GARRE1 risk. This graph shows the Tower phonetic decoding raw scores on the y-axis and the age in months since the first test date for each child on the x-axis. You can see that the initial intercept effect at the beginning of the grade 1 test maintained across the trajectories that were analyzed. You can see that the confidence intervals create kind of a bell shape at the end of these graphs, and that indicates the lower population, so the measures become less precise with the fewer data points there. Here is a similar graph for Woodcock-Johnson passage comprehension. The difference in these groups is present especially at the beginning. The convergence of the confidence intervals near the end of this graph, again, could indicate just the lower number of scores in that outer range beyond 45 months. Finally, here is a representation of the GARRE1 risk factor predicting the rate of CTOP blending words acquisition. You can see that there are differences initially present at the start, where the no risk group is outperforming the risk group, but these effects start converging over time, with the risk group having a steeper incline or a rate of acquisition than the no risk group. Overall, we saw that our growth curves fit the shape that others have found in previous studies, that decelerating growth curve. This is only the second study to look at the role of GARRE1 in reading ability, and the first to look at a wide breadth of reading skills and inclusion of several key covariates in our assessment of GARRE1. Unlike Pang et al. In 2019, we did not include vocabulary or other reading domain-specific predictors of reading measures. In addition, our language variable was somewhat nonspecific in terms of the extent of other language exposure that occurred at home. For example, were the children indicative of an ESL population, or was there a bilingual home environment? We also have only looked at GARRE1 within the NHLP and grad samples. A good next step is to investigate in further detail the nature of this relationship between GARRE1 and so many reading measures. We could look at domain-specific covariates of reading, or start to do longitudinal causal modeling from GARRE1 to component skills like RAN to reading comprehension to look for mediating effects. Finally, I would like to further investigate some time by genetic interactions that may be specific to the time that each reading assessment was performed, for example, at the beginning or the end of the school year. Thank you so much for listening to my presentation and my dogs. I look forward to speaking or corresponding with you if you have any further questions. I can also generate any graphs you would like of specific covariates and with outcome measures if you'd like to see more visuals of the results. I hope you have a wonderful rest of your conference, and thank you so much for joining me.